Structure of polynucleotide chains. Structure of polynucleotide chain. Let us recapitulate the chemical structure of the polynucleotide chain DNA and RNA. A nucleotide has three compounds: nitrogen base, pentose sugar, ribose in case of RNA, deoxyribose for DNA, and phosphate group. These are two types of nitrogen bases: purines. Adenine guanine is a purine, and pyrimidines cytosine and uracil and thymine is a pyrimidine. Figure 10.1. Figure 10.1. Purines and purines and cysteine are common to both DNA and RNA, whereas thymine is present only in DNA. Uracil is present in okay, whereas thymine is present only in DNA. Purines and cysteine are common in both DNA and RNA, whereas thymine is present only in DNA. Uracil is present in RNA at a place of thymine. A nitrogenous base is linked to the pentose sugar through a N-glycosidic linkage to form a nucleoside, such as adenine or deoxyribonucleic, such as adenosine or deoxyadenosine, such as adenosine or deoxyadenosine, and gynosine or deoxygynosine. Gynosine. తర్వాత cytidine, okay, cytidine or deoxycytidine, thiamine, okay, thymidine or deoxythymidine. When a phosphate group is linked to 5-OH of a nucleoside through phosphoester linkage. When a phosphate group is linked to 5-OH of the nucleoside through phosphoester linkage, a corresponding nucleotide or deoxynucleotide depending upon the type of sugar present is formed. Two nucleotides are linked through 3 direction to 5 direction phosphodiester linkage to form the dinucleotide. More nucleotides can be joined in such a manner to form a polynucleotide chain. A polymer, okay, thus a polymer formed has at one end free phosphate moiety, at 5 end of the sugar, and which is referred to as the 5 end of polynucleotide chain. Similarly, at the other end of the polymer, Similarly, at the end of the polymer, the sugar has a free 3-OH group, which is interfered to as the 3 end of the polynucleotide chain. The backbone in the polynucleotide chain is formed due to sugar. And phosphate. The nitrogenous bases are linked to sugar moiety project. From the backbone. Figure 10.2. In RNA, every nucleotide residue has an additional OH group. Present at 2 position in the ribose. Also in RNA, uracil is found at the place of thymine. Also in RNA, the uracil is found at the place of thymine. 5-methyl uracil, another chemical name for thymine. 5-methyl uracil, another chemical name of the thymine. DNA as an acidic substance present in the nucleus was first identified by the Friedrich Michscher. Okay, Friedrich Michscher. Okay, Friedrich Michscher in 1869. DNA as an acidic substance present in the nucleus was first identified by the Friedrich and Michscher 1869. సో డిఎన్ఏ అనేది ఒక అసిడిక్ సబ్స్టాన్స్ అనేది ఫస్ట్ కనిపెట్టినది ఎవరంటే ఇతను ఫ్రెడరిచ్ మిక్చర్ ఇన్ 1869. He named it nuclein. However, due to the technical limitations in isolating such a long polymer intact, However, due to technical limitation in isolation, such a long polymer intact, the elucidation of the structure of DNA remained exclusive for a long period of time. The exploration of the structure of a DNA remained exclusive for a very long period of time. So it was only in 1953 that James Watson and Francis Crick, based on the X-ray diffraction data produced by based on the X-ray diffraction data produced by the Maurice Wilkin and Rosalind Franklin. Wilklin and Rosalind Franklin proposed a very simple but famous double helix model for the DNA, for the structure of DNA. Okay, James Watson and Franklin Crick were based on the X-ray differentiation data produced by the Maureen Wilson and Rosalind Franklin proposed a very simple and famous double helix model of the structure of DNA. So one of the hallmarks of their proposition was based okay. So one of the hallmarks of their propositions was base pairing between two strands of polynucleotide chain. So base pairing between the two strands of polynucleotide chain. However, this proposition was also based on the observation of the Erwin Chargaff. Okay. However, these propositions was also based on the observations of the Erwin Chargaff that for a double stranded DNA, the ratio between adenine and thymine and that between guanine and cytosine are constant and each equal once. Okay, and each equal once. The base pairing confers a unique property to the polynucleotide chains. The chains are complementary to each other, and therefore, if the sequences of the bases in one strand is known, then the sequence of another strand can be predicted. Also, if each strand from DNA, let us call it parental DNA, act as a template for synthesis of the new strand. The two double stranded DNA, let us call them daughter DNA, produced would be identical to the parental DNA molecule. Because of this, the genetic implications of the structure of DNA become very clear. Because of this, the genetic implication of the structure of DNA becomes very clear.